Okay. Hello everyone. Am I audible now?
Yes, sir.
Yeah. So, welcome to the fifth week problem solving session. So, in this week we will discuss about the week 5 lecture content, some topics from the week 5 lecture contents, and you were asked to uh, you can ask if you have any doubt on that. And uh, like other sessions, we will also divide this session by two parts. First, we will discuss the assignment questions, and then we will decide, and then we will discuss some selected topics from this week's lecture content. Okay, so I will present my screen now. Can you see my screen? PPT?
Yeah, sir.
Okay. Okay. So, so the PPT is visible, right?
Yeah.
Okay. Okay. So this week 5 lecture content was about the meiotic cell division. So uh, this lecture content discuss about the meiosis cell division. So we will discuss some topic about that, and I think someone is in the queue. Yeah, 10 people has 10 people have been joined, so I think I can proceed with the discussion. Yeah. So this week 5 lecture content was about meiosis cell division. So we'll discuss about the meiosis cell division a bit, and also it also contained the nuclear import export, nuclear port complex, that will also be discussed in the this week like problem solving session. So the first question, the diploid. So the question is asking that the diploid chromosome number in an Indian elephant is 56. What will be the chromosome count in the elephant sperm cell? So, who wants to answer for that?
28.
28. Why 28? Why not 56?
Because the sperm contains haploid number of chromosomes, that is 28.
Okay, so it has been resulted from yeah, so it has been resulted from the meiosis cell division, right? Sperm cell. Yeah, that's why the chromosome number has been halved, 28, and the chromosome is no longer diploid, it is haploid, right? So the answer is 28. Okay. So next question is, which of the following statements is correct about meiosis? First is, meiosis begins with diploid parent cell. Meiosis results in 4 daughter cells. Meiosis is required for sexual reproduction. All of the above. So first option is right or wrong? Meiosis begins with a
Right.
diploid.
Yes, right.
Because it begins with a diploid twin mother cell, right? So it is right. Second option, meiosis results in 4 daughter cells. Is it correct?
Yes, sir.
Why 4 daughter cells?
Because in meiosis 1, the homologous chromosome
Uh-huh.
separates, and in the meiosis 2, the sister chromatid separate and performs 4 daughter cells.
Yeah, so 2 plus 2, 4, right? That's why 4 daughter cells. Third option is meiosis is required for sexual reproduction. Is it true?
Yes, sir. For gamete formation,
Yeah,
then
for the sexual reproduction you need to form gamete, and gamete can only be found by meiosis. Haploid. Okay, so all of the above are the answer, right? Yeah, so answer is the all of the above. Okay. Third question is meiosis 1 leads to formation of how many daughter cells? Meiosis 1 only. How many?
So two.
Two daughter cells. Anyone else want to comment for that? He told two. Is it true? Anyone else?
Yes. Yes, sir. Two.
Yes, yes. So meiosis 1 leads to formation of two daughter cells. Yes, it is true. Next question is recombination during meiosis leads to new combination of alleles. Is it true?
Yes, sir.
Yeah. Genetic diversity.
Yes, sir.
How
Due to
genetic?
recombination of
How genetic?
due to recombinations, sir, there will be expression of some genes and some genes will be suppressed. Like if mother have some genes and father don't, then something will be expressed and some genes will be repressed.
Okay. Yeah. So the reason of the genetic diversity is specifically is the crossing over, right? Yeah. So crossing over between which two chromatids? In meiosis one, in the prophase stage, non-sister chromatid, right? So there will be alignment of two homologous chromosomes. The homologous chromosomes will be duplicated, and the non-sister chromatids of each homologous pair of chromosome will exchange their sister chromatid, right? So that's how they will get the genetic diversity. And formation of four genetically unique daughter cell.
It's also true, sir.
Yeah, because four, it will give rise of four cells, right? Four progeny. Yeah. So the answer is the all of the above. Anyone have any problem for the last four questions?
No, sir.
No, right?
No.
Okay. For the fifth question, which of the following is incorrect about gametes? What are gametes? And how the gametes can be formed by mitosis or meiosis?
Gametes can be formed by meiosis.
Meiosis.
And gametes are the, I mean,
Gamete. What is gamete?
they are the like haploid set with haploid set of chromosomes.
Haploid, right?
Yes.
Yes. Yeah. Yeah. So when there is so suppose there is for fertilization of the gametes you need one female ovum and male sperm, right? So both of them are haploid, right? So the after fertilization the zygote it will form, so that will be 2n or n, diploid or haploid? Sorry?
Diploid.
Diploid. Diploid, yes, right. So gametes are an organism's reproductive cells. Gametes are on organisms' reproductive cells. Is it true?
No, sir. Every reproductive cell cannot be gamete because there are cells which divide and forms like gamete. So every reproductive cell cannot be gamete.
No, they are not telling that every reproductive cell is gamete. They are telling about gamete. Gametes are an organism's reproductive cell, right? Is it true or false? Gametes are the organism's reproductive cell. It is true?
It's true, right? Gametes are organisms.
Yes, it is true. It is true. Because they are not telling that all of the organism reproductive cells are gametes. They are not, the statement is not stating that. They are saying that gametes are organism reproductive cell. It is true. And what about the second option? Gametes are haploid cells. True or false?
So true. Yes, they are.
True. Very good. True. Third is gametes are produced by mitosis. Is it true or false?
No. False. Meiosis.
Meiosis. Gametes are produced by meiosis, right? Yes. So in the meiosis, as you know that there is meiosis 1 and meiosis 2, right? So for gamete production, you need to separate the sister chromatid, right or wrong? For
Yes, sir.
for gamete production, you need to separate the sister chromatid. So in which meiosis the sister chromatid separation happens?
Since meiosis 2,
Meiosis 2 and meiosis 1, what happens? Which separation?
sir, homologous chromosome separates.
Homologous chromosome separation. Yes, very good. I think so. Yeah, so homologous. Separation of the homologous chromosome. Okay, the fourth. So the gametes are produced by mitosis is the wrong option, right? And the last option is male and female gametes are also known as sperm and ova, respectively. Is it true?
Yeah, sir.
Male is sperm and female is ovum, right? Yeah. So the option is gametes are produced by mitosis. This is the incorrect statement about the gametes. Okay, next for question 6. That which of the following best defines zygotine? So, what is zygotine?
So it is a stage in prophase 1 of
Prophase,
meiosis 1.
yes, meiosis, prophase 1 of, prophase 1 means meiosis 1, right? Prophase 1 of meiosis 1, this is the stage. So, let me just go through some slide here. So, if you will follow the zygotine diagram of this meiotic prophase 1, so what can you see in zygotes? In zygotine, after leptotine, what changes has been occurred in the zygotine?
So pairing started, synaptonymal complex forming started.
Yeah, synaptonymal complex forms it has started forming, and pairing of the homologous chromosome started, right? Pairing means they are actually begins to align, right? I mean, one pair of homologous chromosomes, they have started to align, because for the homologous recombination in the crossing over step, in this step you need to align, you require to align the one pair of your homologous chromosome, and they should be aligned, they should not be up down and side, they should be properly aligned. That's why then only the proper crossing over can be occurred. So in zygotine, basically the synaptonymal complex will be formed and the pairing of the two homologous chromosomes will be occurred. So let's go to the question. Which of the following best defines zygotine? Pairing of the homologous chromosome. So it is true, right?
Yeah, sir.
Tetrad formation is not true, of course. Crossing over is true or not?
No, sir.
But crossing over, it also, I mean, it also takes place in
In later, not in
zygotine,
not
right?
Zygotine.
So in which phase it occurs? Sister crossing over? Yeah,
Sir, diplotin.
not
No, sir, no. Pachytine.
pachytine. Yes, pachytine. Yeah, and termination of chiasmata, this is also not possible, right? Because chiasmata will form later in actually diplotin. You can see you will be able to see the uh, chiasmata, right? So that will start appearing from the diplotin. So you can't see the termination of the chiasmata in zygotine. So the first option is right, the pairing of the homologous chromosome. Answer is the pairing of the homologous chromosome. Next question is, as at which stage the synaptonymal complex disassembly occurred? At which stage?
So diplotin.
Yes, diplotin. So if we will again visit the diagram, you can see that in diplotin, so in leptotine condensation, zygotine pairing, pachytine crossing over, and in diplotin, see up to this region you can see this synaptonymal complex present. Here also present. Here it just started, and here is the it is present in pachytine, but In the diplotin, the synaptodimal complex starts to disappear, and due to this the chiasmata are visible, because here chiasmata is not visible because it was this it was shielded by the, or it was not visible due to the presence of this synaptodimal complex. So in the diplotin, the synaptodimal complex will start to disappear. Yeah, so the answer is diplotin. Anyone have any problem or any question up to that?
No, sir.
Okay. In budding yeast, IME2 acts as a what? Meiosis specific transcription factor, meiosis specific phosphatase, meiosis specific inhibitor, meiosis specific kinase. Phosphatase, transcription factor of what? Kinase.
So transcription factor.
Not transcription factor actually. Can you
It's
remember?
kinase.
Yes, it is
Sir,
kinase. It is kinase. And what is its substrate? Do you know? Substrate of IME2? Substrate is IME. Sorry.
MEI2.
Substrate is IME1. Okay, IME1 is the substrate for IME2. And one more thing that, so about the third option, in this telling the meiosis specific inhibitor. So do you think that IME2 is a inhibitor of meiosis or activator? What can it be?
Sir, activator.
Activator, because activator, yes. So IME2 mediated phosphorylation of IME1 is activate. This event activates the meiosis process, right? Yeah. So meiosis. So the budding yeast IME2 acts as a meiosis specific kinase is the right answer. Which of the following is a transverse filament protein present in the synaptodimal complex? Remember about the synaptodimal complex? Among these four options, which is the part of the transverse filament protein? ZIP1, RED1, GMC2, HOP1. Which
So
one?
ZIP1.
ZIP1, very good. ZIP1 is the member of the transverse filament, which is found to present in the synaptodimal complex. Yes. Answer is ZIP1. Okay, now some question about nuclear import export. So in the RAN-GDP complex, hydrolysis of GTP to GDP occurs during nuclear import inside the nucleus, nuclear import in the cytoplasm, nuclear export in the nucleus, nuclear export in the cytoplasm. So
Export in the cytoplasm.
export in the cytoplasm, right? Yeah, yeah. And we will discuss about the nuclear import export mechanism for by sharing one example. Here we will see it in detail. Okay, so the answer is nuclear export in cytoplasm. So now just let me know if you have any doubt about the question we have discussed till now. 10 questions we discussed. So do you have any doubt for that? Or any point you need some clarification, you can ask me if you want.
Sir,
Yeah.
can you please explain sumylation? It is little bit we don't know about it, but ma'am used in lectures about the term.
Okay, in general, if you want to ask the sumylation is the sumo stands for small ubiquitin like modifier. Okay, so it is also so this sumylation is also a post translational post translational modification, right? So can you give an example for the proteins which is known to be sumoylated?
So P14K, like I'm not sure, but like this.
P14K. I don't know also about that. Maybe it may be, but one of the common example is PCNA, right?
Yes, sir. PC.
PCNA is also known to be simulated. So this simulation also, if you will see the machinery for the simulation, it is exactly same as the ubiquitin, how the ubiquitination occurred. Okay, because here also you will find the E3 ligase, who will transfer the sumo to the sumo protein to the substrate, and there is a SMC complex called SMC56 complex. In this complex there is a protein called NAC2. In this NAC2, it has been found that it has some sumo ligase activity. So sumo ligase activity, there is nothing to be confused, because it is just like, that's why it is called also that ubiquitin like modifier. Then the process of simulation is also similar to the process of ubiquitination.
So like this one also have ring and hex domain like E3 ligase.
Yeah, ring
This one.
domain it has. I don't know about the hex domain, but ring domain basically it has. And the same
But it's
thing, E3, E3.
one thing I read about in Google that it doesn't promote the protein degradation like
No, 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 no. That is the thing. It does not promote the protein. I mean proteasomal degradation. Basically, it will act to recruit something. It will just, if any of your protein is simulated, that means you can get a guess that this protein, the simulation, the objective of the simulation of the particular protein is to interact with someone. That means it can recruit someone, right? So
like signaling something.
yeah, signaling something, or if if it needs to recruit something in the, in the chromosome, then also it is helpful. So that does
So
not.
how does our cell differentiate between the ubiquitinated protein and like simulated protein? Like there is ubiquitin chain in both of cases.
Yeah, but it's smooth. It is ubiquitin like, but not ubiquitin. The sequence is not same. So
Okay, okay,
if
sir.
the sequence is not same, then cell can of course recognize it. So
Okay.
ubiquitin, basically, see, if your protein is ubiquitinated, okay, then your protein can interact a protein which has the ubiquitin binding domain, or UBD, right? So that ubiquitin binding domain can only recognize if your protein is ubiquitinated. If your protein is simulated, then that ubiquitin binding ubiquitin binding domain, UBD, cannot recognize it, because sequence is different. So that's why it is important that although they are similar, but they are not identical, right? So the UBD cannot bind to the sumo, or in the proteasome, as you know that the proteasome can recognize by the polyubiquitin chain. So in, so that proteasome can only recognize the polyubiquitin chain. It will not recognize the simulation. That's how, although it has some uh, similarity, but still they are not the same, or cell can actually differentiate, because there is a difference in the sequence. You got it?
Yeah, sir. Got.
Yeah. So uh, in the range, we just discussed it right now. Okay, so any more doubt? Any more question you want to ask?
Sir, role about telomere in meiosis,
Telomere in meiosis.
like in prophase one.
Uh, I mean, which role you were talking about?
Like ma'am just told something about like telomeres, um, nuclear envelope goes to the nuclear envelope and rotates in any position and accumulates in a specific place.
Okay, see, there is the important thing is attachment or tethering of nuclear membrane and telomere. Okay, that is also important to maintain the chromosomal structural organization. Okay, so yeah, so for that, and that chromosomal, I mean the telomere attachment to the nuclear membrane is actually required for any kind of spatial organization of the whole nucleus. And I think that's how it will affect the, meiosis.
So, like when they are condensing the chromosome,
Huh.
they have to like specific space for every chromosome, so
Huh.
they
Huh.
recognize it by telomeres,
Yeah.
and places every chromosome in the place required for them to separate.
Hmm. Hmm. Yeah.
So they actually bind with the nuclear envelope, or just any kinesin like protein
No,
or
no, no,
monomer
no.
protein.
Telomere directly bind to the nuclear membrane, and this event is called as telomere tethering to the nuclear envelope. There is some nuclear envelope protein to which telomere can bind. So telomere
Okay.
should be attached to the nuclear membrane. That actually that will help to give the proper spatial organization of the telomere. So that is required for that. And as you know that yeah in budding yeast, because we are talking in the context of the budding yeast. So in budding yeast, the nuclear envelope will not completely be broken down. So this attachment will be there. Okay.
I have one specific doubt.
Yeah.
Like in meiotic process, like when homologous recombination occurs,
Uh-huh.
there are some proteins like double strand break occurs, then arrangement of chromosomes, and then formation of that strands
Yeah,
occur like
yeah,
that.
yeah. Strand infection.
But we also know that in double strand break there are in your our DNA management system there are Reg A, Reg B, Reg C protein
Yeah.
which conducts this double strand break to
Repair.
uh, repair.
Uh,
So how does this happens, like in this stage other proteins act on this, and like when double strand break occurs in nucleus for like in S phase or anything, then the Reg A, Reg B, Reg C protein recognize it.
yeah, yeah, yeah. Because see if you are if you are going through the meiosis condition, right? Then the total protein pool of your cell will not be same as the if you are going for the mitosis, right? So if for that's why the double strand, when in any textbook when you will read about the downstream double strand break repair, so you will encounter two different double strand DSB repair system. One is your the normal, where the which is actually the part of the DNA repair, and the where the that 51 foci will be formed, and then sanitation everything will be done. So that is one part, and the another thing is the crossing over in the meiosis. So for crossing over, there are different pool of protein will be present, and in the S phase DSB or any kind of DSB, then they have some specific protein, like as you told that Reke foci will be formed, and Reke actually in, I think, bacteria, right? Prokaryotic. Reke,
Yes.
yeah, Reke, I think, and that is a part of DNA repair, DNA repair machinery, right? But in yeast or eukaryotic system, you will find the RAT51 foci formation. When there is revolution break, and then after that what happens, there is streaming of the 3' right, and this streaming and the streamed 3' at this position, the RAT51 foci formation, which will lead to the strand invasion, right? So that's why I think you were talking about Rek A, Rek B. Rek A, Rek B is not part of yeast or eukaryote, that is part of bacterial system. Okay, and that is true when the bacterial, there is double strand break, and you need to repair the double strand break, then your like like A, B, C, that foci will be formed. But in case of yeast, there is that 51 foci formation will be formed, and then the homologous recombination will be occurring. Okay.
So like that every event happens in meiosis also regulated by cyclins and any phosphorylated things.
Yeah, yeah, of course. In meiosis, like as I already discussed with you, that I am one of the specific kinase is IME2 kinase, right? So this also activates the meiosis because it phosphorylates the IME1, which is an inhibitor for meiosis. So this also have a CDK like activity because phosphorylation of this kinase is actually helping to promote the meiosis. So that's why. So this is one of the example. IME2 is one of the example like CDK in, as you saw CDK in mitosis. Similarly, IME2 here is a example for example of a kinase which can promote the meiosis. You got it?
Yeah, sir, got it.
Yeah. Anyone questions? Anyone have? Okay, let's then move on to the some selected topics from your lecture content. So let's first discuss about the meiosis division. This is the normal meiosis division. So what basically happens in prophase? You can see prophase the chromosomal condensation will be occurred. Again, the condensation is only specific for the this division phase. So in prophase, you can see the and already the S phase has been occurred. So so each DNA has been duplicated. You can see here. So after the duplication, the thing is this is a diploid cell, right? This is a diploid cell. Yeah. So in the diploid cell, you can see when in the diploid cell, the first thing in the prophase which has to be occurred is the proper alignment of the homologous pair. These two violet are homologous pair, these two red are homologous pair. So it is important that in meiosis 1 prophase 1 the homologous chromosomes should be aligned to each other here. And one of the major reason of the proper alignment is the proper crossing over or proper cystochromatic exchange. So here you can see that for the prophase the homologous pair of the chromosome has been aligned with each other. After metaphase, we can see the formation of the spindle pole and the projection of this spindle fiber from the spindle plate and the attachment to the kinetochore that you can see. And now you can see that the pulling this entire two chromatids are pulling this end, and in this entire two chromatids are being pulled by this end, right? So that is the difference from the mitosis, because in mitosis the two sister chromatids should be separated, right, by the spindle fiber. But here that entire these two sister chromatids are being separated. Actually, the homologous pair of this sister chromatid is being separated here, right? Similar thing for the pink pink uh, pink homologous chromosome pair. So that is the meiosis 1. So after meiosis 1, what will be happen? After meiosis 1, you will find one this and one this, because its homologous pair has been segregated. Its homologous pair has been segregated. So when if cytokinesis will be occurred according this plane, then you can see that so if the cytokinesis occurs in along this plane, you can see that one of the homologous pair and one of the homologous pair, but they are not homologous to each other, but their homologous pair has been separated. Here also same thing, here also same thing. So ultimately it will result to a cell which contains this and this, a cell which contains this and this. And in the meiosis 2, now it is the same like mitosis. So in the meiosis 2, the two sister chromatids will be separated, and after the separation you can see that uh, if you can see that here you will find that after the division the cell will contain these two, right? So now these two sister chromatins will be separated, two blue sister chromatins will be separated, and two pink sister chromatins will be separated. So after the separation, cytokinesis will be occurred along this plane, along this plane, and now it will give rise to a haploid, because and don't think that if and don't think that they are aligned towards each other. That does not mean that they are 2n, because they are not homologous. See, this homologous chromosome has been already taken out, so they are not homologous. So the meiosis 2 non-homologous. So in the meiosis 2 we can find the haploid copy of chromosome, because there is no homologous pair for this and there is no homologous pair for this. So in the all progeny we will find the haploid state. So this is the meiosis cell division. So anyone has any doubt in this?
No, sir.
Okay. Meiotic prophase 1. Okay, so as we already discussed it, that the stages of the meiotic prophase 1, you can see that in leptotin, it starts from the leptotin. In leptotin, the chromosomal condensation will be occurred, and after that the chromosomal, now after the making the chromosomes condense, now you need to, in zygote you need to pair your homologous pair of chromosome. So now you can see that these two are the homologous chromosome, this and this. So now after the duplication, now they has to be aligned with each other, so that these two non-sister chromatids can exchange their so can exchange their genetic material by the process known as crossing over. So and and that is taken place in packaging. So in zygotic chromosomal pairing occurring, so in the zygotic chromosomal pairing occurs, and synapsis begins, because there is just loading of the synaptonium complex here, and also chromosomes are full synapse and crossing over begins in the packaging stage. And you can see that still up to the packaging stage, the synaptonimal complex is still here, but when it will reach to the diplotin, then the synaptonimal complex will be lost. They will start to disappear, and you can see that after the disappearance of the synaptonimal fluid, you can find the chiasmata here. So so after that diakinesis will be occurred. So now so this is the entire description about the meiotic prophase 1. Anyone has any doubt in this meiotic prophase 1?
No.
Okay, so now let's look about the synaptonimal complex, which I told you that it was formed during this stage, when the zygote stage, when there is a pairing of the homologous chromosome has been occurred, then the formation of the synaptonimal complex is formed. So synaptonimal complex, if you find the structure of the synaptonimal complex, you can see that there is three part is visible here. One is central element. This is a central element, pink one. There is there is transverse element. This and this is the transverse elements. After that, there is this is the reaction of the chromosomal axis, and here you can see that chromatin loops. So this is the meiotic prophase. where the synaptonuclear complex of the binding is formed. So now, as I think we are discussing about the homologous recombination, and homologous recombination is taking place in the crossing over. So as you told, so as you asked me about the question that what is the difference between crossing over homologous recombination and non-crossing over homologous recombination? Was the same question did you ask me? Crossing over
Yes,
and
sir.
yeah. So how these processes are different, right? So that we will see here. So suppose there is a double strand break, okay. So now see, double strand break can be occurred in the stage of crossing over in the meiosis, and it can also be occurred if your DNA has a double strand break due to the DNA damage, right? So these are two possibilities. So the same pathway for these two double strand break is first is the rejection of this 5' end. This is the 5' end. Initially it was like this, but after the trimming it is like this. Same here. This is also the 5'. Yeah. So here you can see that at the 3' end there is, you know, trimming has been occurred, and now there is a single-stranded region here. So now after that this strand will be invaded, right? Invade to the to its homologous chromosome, because of course because it needs to have a template for continuing the continuity for keeping the continuity of the reaction. So for that it needs someone by help of which it can originate two different uh, pathways for two different purposes. So here the first step is same, double strand back repair, and in the there is a rejection or exonuclease activity at the 3' end. That's why it has been trimmed. After the trimming, the strand inversion will be taken place. This was the yeah. So this was the initially the blue one is this DNA chromosome, and there is another red one chromosome. Okay. So in red one chromosome is actually homologous to the blue chromosome. So now this strand will invade and use this homologous pair strand as a template, and it will synthesize the DNA here. And similarly, uh, this region, this this part this part will come to close to you and use a template with this, and after that this 3' end will be amplified. So this is the double strand homologous recombination in the crossing over in the meiosis specifically. But when it comes the thing of non-crossover homologous recombination, then you can see that first of all it is same, the incorporation of the double strand break, and then trimming of the 3' end, and then strand invasion. But after that, in this pathway HDSA, after that there will be dissociation. Okay, the strand has been invaded, but after that the strand will form and the strand will be dissociated from here. So after the dissociation, this strand, and now before the dissociation, this strand has it acts as a primer, and it can form the DNTP here, like by extending the 3' band. Here also you can see that after DNA formation, it will be dissociated along with this red lines. So after the dissociation, annealing should be done. In the annealing, this region will, and now you can know that this region is complement to this region. So this region will anneal here, and yeah, so this region will be annealed here, and this portion will be extended to make the double strand DNA. So if you have any doubt, meiotic prophase homologous recombination, tell me. Is it fine? Is it fine?
Yes.
Okay. Yeah, so now did you get it? That how that how it is different between the meiosis homologous recombination and the DNA repair homologous recombination.
Yes, sir.
Yeah, so did you able to find did you able to catch the spot where the difference lies between this you I think know DSB repair, but this is somewhat uh, tricky, right? Because this DSBR, it is this pathway is followed in the crossing over in meiosis, but in HDSA that is a non-crossover pathway. So so up to this, the presence of the DSB and the strand streaming, and then the invasion, up to that the thing was same. But you can see that the difference is that here the stable strand, this is stable strand invasion, because because it will not be dissociated. Same was done here also HDSA, but here you can see that it is stable, it is not getting dissociated, it will complement to each other, so it will anneal here, and it was and it acted as a primer. DNA synthesis was going on, and after that only uh, this egg shaped structure formed, but this region never dissociated from here. If you will see this figure, if you will see this figure, this region will never never dissociated with this, but this region was shown to uh, you know this region was shown to anneal to this strand, and this acted as a primer, and this acted as a template for this synthesis. But here the yeah, but here the difference you just see here, just that here also the same thing, the the strand extension was going on here gradually, but after some time the strand invasion was not, or the strand extension here was not stable, so it was dissociated from here. See, some length was extended, but after that it has been dissociated, and then again it will anneal to the here, it will anneal to here, so it will anneal to here, and yeah, and then the remaining strand extension will be occurred. So both of the cases the homologous recombination is being taken place, but the way of the how the HR pathway here is being utilized here, that is different. So did you find the catch here, the difference?
Yes, sir. But sir,
Yeah.
what is the full form of HDS?
SDSA, I think single strand dissociation, strand anything something like that. Wait, just let me. I forgot the full name. Wait. Yes.
And sir, did DSBR forms the homologous? Sorry, sir. I mean the holiday junction, the X-like structure.
Yeah, that is the holiday option, and that has to be. There should be resolution of the structure, right? Resol- Otherwise, this shape will be permanent here. So you need to do the resolution here. For that, it is important.
After it, chiasma is formed, right? X-like structure.
Yeah. After the crossing over is complete,
Sir.
then I think it diplotin, right? In
Yes.
diplotin, the complex will be disappear, and we can see the chiasmata when the entire crossing over has been completed. Then only. Okay, so we got the meiotic process homologous recombination. Two kind of homologous recombination we found. That is the reason. Okay, so now nuclear envelope. So what is nuclear envelope and what can be its role? Nuclear envelope role. Okay.
Sir, it separates the like chromosomes, then the other cytosol from like different proteins that
Yeah.
can
Yes.
start everything.
Yes. That's why it is envelope, right? It is a just protection. It is not let the nuclear content mix to the cytoplasmic content. So this is the boundary. Here you can see that. So about the nuclear envelope, you can see that there are two nuclear membrane you can find: inner nuclear membrane and outer nuclear membrane, and this inner and outer nuclear membrane are separated by the lumen. ER means endoplasmic reticulum. And then you can see that the lamin B receptor, it interacts with the B type lamin. This is the lamin. If you remember, in the last class we discussed about the nuclear lamina, where we showed that how nuclear, lamin protein phosphorylation will help the mitosis. So here the same thing, lamin is there. This is the lamin protein. So the lamin B receptor interacts with lamin. Lamin is the sticky things with the lamin and chromatin associated heterochromatin protein HP1 here. So it can interact with nuclear lamina as well as the chromosome by this HP1 protein. And members of the LEM, the lamina associated protein 2, like LAP2. LAP2 is a lamina associated protein, right? LAP2 bind to the lamin. LAP2 sequence, LAP2 bind to the lamin and interact with the chromatin through the barrier to the BAF, and it interacts the chromatin through this BAF. Like LBR here, it also interacts with lamin, and also it interacts the chromosome by this mediator HP1. Similarly, here LAP2 interacts with lamin and interact the chromosome with this BAF uh, BAF protein. Then here is some sun protein. Sun proteins interact with the nesperins in the ONM outer nuclear membrane, thereby forming a complex called link complex, and then establish connection to actin and intermediate filaments in the cytoplasm. Yeah, so this is actually responsible to the connection between actin and intermediate filament. Here you can see there is generation of the projection of the actin or intermediate filament. Now, yeah, so this is the overall nuclear envelope structure. So, does anyone have any doubt here? Any, if I miss anything, is it completely clear now?
No, sir, it's clear.
Okay. Okay. So the next is the nuclear. Yeah. So the next is the nuclear pore complex or NPC. So if you will see the structure of the nuclear pore complex, you can see that nuclear pore complex which forms between two nuclear pore, right? Nuclear pore is because say nuclear membrane is not intact, right? This is a discontinuous membrane. So there will be generation of the pore like this in the nuclear membrane. So and in this pore, this is the nuclear membrane and this is the nuclear pore complex. So in this pore, the NPC complex was embedded, and the structure of the NPC complex is there is a central pore in the middle, there is cytoplasmic ring which extrude to the cytoplasm, there is a nuclear ring which extrude to the nucleus, and there is nuclear envelope. This is the nuclear envelope and this is the nuclear pore complex which has cytoplasmic filament ring facing towards the cytoplasm is a central pore, and there is nuclear ring and nuclear basket which is facing to the nucleus. So that is the nuclear pore complex formation, and I will just discuss about the nuclear import process of a protein TRSP1. So now you tell me if you have any doubt after this. You have any doubt? You got the why it is called nuclear pore complex? Because it is situated just between two pores. That's why it is called. I mean, it is situated in the pore. This is the envelope. This is the envelope, and this is the pore. That's why it is known as nuclear pore complex. Any doubt in this? You can let me know.
No, sir.
Okay. And also I forgot to mention that is nuclear basket here, in which is facing to the nucleus. Okay. So next is the nuclear import of TRSP1. Okay. So what is TRSP1 disease?
TRSP means transport protein complex.
TRSP1 is a transcriptional regulator. Okay. So if it has now see how its nuclear import is important. It has this gene encodes a zinc finger transcription factor which contains two region with basic amino acid, this motif which is NLS1 and the another motif which is NLS2. Now you tell me what is NLS, what is NLS? What is the full form of NLS? Full form for NLS? Any idea? And why it is required? Yes, please.
We forget it.
Okay, no problem. The NLS is nuclear localization signal. Okay, if you want to import any protein inside the nucleus, you have to have the NLS sequence, because this NLS sequence will be recognized by the nuclear importins, And as you know that proteins are responsible to mediate the nuclear import of a protein. So to get inside the nucleus, you need a protein, and without NLS you can't get the interaction with the protein. That's why the nuclear localization signal is required for, yeah. And that is actually, there is no hard and fast constant sequence for the NLS. Many proteins have many different NLS, but the one thing is that you should have some arginine rich residue in your NLS sequence. Like if you will follow it, you can see that there is L triple R. Here you can find 5R, so 5R arginine. So this is a common, this is a signature mark for being a sequence to be, or a motif to be NLS. Okay, so now this gene so now this gene encodes the transcription factor, and to act as a transcription factor, you know that you need to go inside the nucleus, otherwise you can't do your job. So if you have mutation in your NLS sequences, then you can't get to the nucleus, and you you can't do your role as a transcriptional regulator. So that's why you will so due to this mutation, there is a possibility that you will develop disease, because this protein is not able to do its work. And deletion or mutation of the TRPS1 gene leads to the disease trichorhinophalangeal syndrome. So it is basically a problem for bone and joint. So and it the reason of this disease is the deletion or mutation in this TRPS1 gene. And as I already told you that it has NLS, and for doing its activity, it need to go inside to the nucleus. So if you will block this nuclear import, okay, by mutating this NLS sequences, so if you will block the import of this TRSP1, then it can't play its role as a transcriptional repressor. So that is important. As the example is surrounded, because as the example is based on the TRSP1, so I just wanted to give you a short note about TRSP1. So let's understand the nuclear import process here. So this is a cytosol, this is nuclear membrane and the nucleus, and this is the nuclear pore complex, right? NPC. Okay. So so this is the NPC. So now see, start from here. Okay, this is the TRPS1 protein, and this has the NLS sequence. Now, as I told you, this NLS sequence can be recognized by the imported alpha. Okay, so the imported alpha will bind to the NLS sequence, and imported beta will bind to the imported alpha. This combination is required. Imported alpha beta dimer, imported alpha beta and bound with your carboprotein. So this combination is required to go inside the nucleus to this nuclear pore complex here. So after going into the nucleus, so this nuclear pore complex, now this combination, your protein and imported alpha, imported beta, so upon coming to the inside, RAN GTP came. So after coming to the nucleus, the RAN GTP came, and the RAN GTP, what it did, it has the interaction with the imported beta, but now there is imported alpha. So RAN GTP will displace the imported alpha. It will displace the protein which is to be translocated to the nucleus, and it will bind, RAN GTP will bind to the imported beta, right? Okay, and now this combination is required for mega nuclear export to go to cytosol from the nucleus through this nuclear pore complex. So this came out from the nucleus, they came out to the cytosol, and after that GTP hydrolysis will be taken place. After GTP hydrolysis, this imported beta will be dissociated yeah, imported beta will be dissociated from this, and NGTP will be a separate thing. Then again in the next cycle, imported beta will bind to the imported alpha, and imported alpha will bind to the NLS of the target protein. Or here the target protein is TRPS1 alpha. Sorry, TRPS1. So and after that the same thing. This combination is required for the nuclear import, and then dissociation of the nuclear alpha. Sorry. Then the dissociation of the imported alpha, and then the export of this thing, and then again the same thing. So that's how the imported alpha beta helps in the nuclear translocation. And RAN GTP is also see RAN GTP. So just by looking at this figure, you tell me that where the concentration of the RAN GTP should be more, nucleus or cytosol? Can you tell me?
So which one, RAN GTP or GTP?
RAN GTP.
GTP is inside nuclear.
Okay.
Nucleus.
And RAN GTP where?
Outside, like cytosol.
Yes. So this gradient should be maintained, right? Then only the transcript, the import export will be occurred. So that is the thing of nuclear import, and here the example was used TRSPOP1. So if anyone have any problem, please let me know regarding this, because this is very, because you need to understand this. Nuclear import of TRSP1 by this machinery. Can you have any? Do you have any problem?
Like sir, in lipid soluble proteins which can directly consider the nucleus to their signaling molecule. So there also this happens like the import or export, or like they can thoroughly pass the nuclear membrane without anything.
No. To pass the nuclear membrane, you need to go through the, this importing.
Nuclear pore complex.
Yeah. Because
so like they also need some kind of import proteins for pass through the nuclear membrane.
Who needs NPC, nuclear pore complex?
Yeah, like hormones, like which cell signaling molecules like lipid soluble molecules, estrogen and hormones.
Yeah, like you are talking about the steroid hormones, right? Steroid.
Yes,
yeah. So in steroid hormones, what can happen? It has their own receptor inside the cytosol, right? And these receptors are required to take the estrogen, or estradiol actually, to take the steroids to the nucleus from the cytosol, right? And this receptor should have the NLS so that it can pass through this NPC, and that's why the receptor for these ligands, like as I told about the steroid hormones, the receptor for these ligands are important, and they should be able to transport throughout this nuclear membrane. So if there is no receptor, so the hormones, because hormones can't have the NLS, right? Hormones can't have the NLS, so they can't pass the here, right? NPC they can't pass. So that's why they need a receptor which is a protein, and probably it has a NLS sequence, and then only the steroid hormones which are known to come inside the nucleus, they can come and do their work. You got it?
Sir, like and sir, like we use protein and enzymes like anything for like any vaccines and anything. So we need to put the NLS for them also to be transported into the nuclear membrane.
Okay, for
like we design some proteins or drug.
Yes.
For transport into the air. So we need to put NLS also there.
Of course. I will ask some question to you also from this slide. Then I think it will be more clear. So if your target destination is nucleus, without NLS, I don't think you can do anything. So you need to put the NLS. Okay.
Sir, did it maintains the protein structure? Like
yes, of course.
So like in ends or in middle we can put anything.
Yes, that is the point. NLS you can put at anywhere. Okay. NLS you can actually put at.
So it doesn't destabilize the protein structure like you told.
Like basically what is recommended, what is recommended, or what I prefer is to put the, if you want to if you want to clone a gene for transporting to the nucleus, then you have to give a NLS, right? So now,
Yes.
yeah, so now what you can do that to import the NLS, you have to do some trial and error, right? You can, so the best thing is, if you want to keep your protein structure as native
Form.
as you can, so for that the best thing is to modify the or the insert the NLS in the loop region, which is not conserved, and who and the loop region does not have any such kind of secondary structure, right? This is a completely loop.
Yes.
So on that, yeah, so on these regions are quietly safer if you will put your NLS there, because it because then there is a low chance that your protein structure will be sacrificed. Okay, so that's why the low conserved region or the loop region, that can be a better choice to put your NLS, right? So now just tell me one thing. Is it possible that a protein? I am asking to everyone. Okay, please try to respond. Suppose a protein is there, and the protein, it has been found that the protein localizes into the nucleus, but the protein does not have any NLS. So is it possible or is it completely hypothetically? I mean, it is not possible. Is it possible, first of all? A protein,
No.
not possible. Can you think a bit that if it is somehow possible without NLS localization? Can you think of any way for that?
Sir, as per I know that only mRNA can pass without any transporter or anything.
No, mRNA for export, mRNA export. That there is also they.
There is no transporter.
No, no, they, yeah, they also require transport. I mean the
For mRNA?
transporter. Yes, yes, mRNA. I will just discuss about the thing mRNA export in the next slide. Then you will see that mRNA also needs transporter. Okay, so now here, if a protein, uh, I'm just asking you that a protein does not have NLS, but still you are getting the protein to be localized in the inside the nucleus. Is it possible? By any way? By I mean anyway. So you can just think about and tell me that is it possible by any chance? See, the protein synthesis is taking place in the ribosome, which is in cytosol, right? So for cytosol, it is coming to nucleus, but it does not have the NLS. How can it be possible? If it is occurring, how can it be possible? Any idea you are having in your mind? And this is a research problem you can be encounter it later also, right? Then you need to think that why is it happening? Although the situation here is completely hypothetical, but I just want to know that whether you can think about that or not. Any input? Can anyone please volunteer for that? Anything you just have in your mind? Or should I tell you?
Yes, sir.
Okay. So you don't want to try more?
No, sir.
Okay, no problem. See, is it possible that suppose this protein does not have NLS, okay? So so it is also possible, right? This protein interacts with some protein which have NLS, and as this protein interacts with the protein which contains NLS, now it can go inside the nucleus, right or wrong? Tell me please.
Yes, sir.
So it is possible, right?
Yeah.
So your NLS, so your if you don't have NLS, then you can interact with a protein who has NLS, and and by interacting with this protein, you can go inside the, because if that protein has NLS, it can interact with important alpha, and then important beta will come, and you will go inside the nucleus. Is it clear?
Yes, sir.
Clear, right? No doubt, right? Everyone I'm asking? Clear? Clear, right? Okay. Second question I'm asking you that a protein that has NLS, okay, that has NLS, but supposedly you saw that the protein is not localized into the nucleus. Is it possible? Again, the same question. Is it possible hypothetically? Is it possible or not? By any chance, is it possible? Despite of
So
having
that it
NLS,
has NLS, but we
huh?
can't see it in nucleus.
Yeah, this has NLS, but we can't see the protein in nucleus. That means import is not happening. So is it possible? That's the reverse case I'm asking you. Ulta.
So like that, due to some interaction with other proteins, the NLS is in like smashed between the
Yes,
proteins and.
very good, very good. Exactly. That can be a reason. That can be a very good reason. Then that if the protein's NLS is being involved to interact with other protein, okay, then what will happen? Then the NLS will be masked, right? NLS will be shielded. So now this NLS is as it is in the interaction surface with the other protein. Now this NLS cannot be recognized by the imported alpha. So now it cannot go inside the nucleus. Okay, got it? Very good.
Yeah,
Very
sir.
very good. Very good. That's how you should.
Are these published?
Huh?
Are these things published?
Not publish. I'm just asking you that whether whether you can think about such
Okay, sir.
situations or not, because see, research is always novel thing, right? You don't know.
Yes.
I mean, everything is not known till yet, right? That's why research is going on. So if someday you will find, you will you will encounter such kind of situation, then you should have the capability to think that what would be the cases, right? And it may be the which I told you about the example. It may be true, but I don't know. It may be true. If you are interested, you can just search.
So like, sir, here there are arginine residues which
Yes.
are positively
What is it,
like
Vijay? Yes, yes.
long charge, so they can interact with negative charge proteins like
Of course, of course, of course. Like aspartate, which has the
aspartate,
yeah, aspartate,
glutamate.
glutamate. Yes,
Yes, sir.
yes. With them, they
And
can
form
interact.
ionic bonds or something else.
Yes, 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 yes. And then the NLS will be hidden, right, or shielded. That will be not accessible for the importing. So it will not be here. Just tell me one thing, that how will you, if I will ask you to design one experiment, that you need to check that whether suppose a protein, okay, TRSP1, TRPS1, in one case you have the wild type TRPS1, and the another case you have TRPS1 but NLS, you have mutated the NLS, you have mutated all the arginines, or you know. So in this wild type and mutant, if I will ask you to measure or to say or to find that whether the wild type or the mutant TRSP1, whether they are accumulated in the nucleus or not, how will you assay that, or how will you design your experiment? That
So
whether
like the
huh?
NLS binds with the specific site of the imporins,
Huh?
like imporin beta and alpha. So
Yes.
I will try to find this position and try to like I will design the proteins like those two, and then I will attach an immunofluorescence or anything with them.
With
So
with
can I
whom?
can
With whom? With whom? With whom?
which are the imporins?
Imported.
Ah, yes, sir.
Okay, you will tag imported. Okay. Yeah, see, but the problem if you will tag the imported is common for every nuclear import of every protein, right? It is common for
Yes.
every protein. So if you will see the imported is going inside, that does not mean that imported is carrying your protein, right? Imported may carry other protein, because
So sir,
imported.
there may be a chance that NLS has different sequence for every protein, like different number of arginines or anything
Yeah,
sequence.
yeah, but the imported
Sir,
is same.
I will design a regulatory protein like which will attach
Huh?
by my protein. I need
Huh?
what?
That.
Then I will attach. I will tag the protein,
Huh?
and when it will attach,
Huh?
the wild type will be immunofluorescence. So,
Okay.
but the regulate but what
Huh?
is not the wild type, I mean changed something or interacted with
Okay.
others will not show the
Yeah,
response.
yeah, yeah. Got it, got it. Yeah, this is also a good option that you were telling that you will find the regulatory protein which interacts with the with your protein, right? So for that, that protein should exist,
Yes, yes.
right? For
Hmm.
that, that protein should exist then only.
Because there is no modification, so it will show fluorescence. But if there is any modifications or anything
Hmm.
interaction happens,
Hmm.
then that will be not the wild type. So I can recognize by this immunofluorescence.
Okay, okay, you can do. But actually, if you if you want to directly measure that whether your protein is imported to the nucleus or not, for that purpose, the best experiment, what, that is the FRAP, fluorescence recovery after photobleaching. So in this thing, basically, you will first photobleach your nucleus. Okay. So now, first of all, you will tag your protein by GFP or any fluorescence tag. Then you will see, so after that you will photo bleach the nucleus. Okay. So after photo bleaching, photo bleaching the nucleus, all the existing these proteins in the nucleus will be all of them will be bleached, right? So you can see them after a few minutes. If you will see that fluorescent signal is being recovered in this region, that means that some of your proteins is coming from cytosol to nucleus. If they are imported, if their importing will be occurred from cytosol to the nucleus, then only you can see the fluorescence recovery in their nucleus after photobleaching. But in your mutant, if your mutant is turned out to be defective, and if your mutant will not come to the nucleus, then after photobleaching you will see that in the nucleus the fluorescence is not able to be recovered, because this protein is nuclear import. I mean, this protein has defect in the nuclear import if you have mutation in the NLS. So due to this problem, it will not be imported, and as the GFP is tagged with there, so GFP will not reach to the nucleus if it will not be imported, and you will not see any fluorescence recovery after photobleaching. So that is a technique or direct technique by which you can do the uh, or rather you can measure that how I mean whether your proteins are, whether your nuclear import of your proteins are affected or not. That can be an experiment. And otherwise also uh, also what you told, that if such kind of interacting protein exists, then you will do the immunosuppression. That is also very good technique. That will also work.
Sir,
Okay. Yeah.
like sir, we can do any part of the cell, like any smaller and very small part of the cell to photo bleach.
Yeah, you can target it in the FRAP technique. I will rather suggest to read more about the FRAP technique.
Like sir, I read about the like the cell membrane. There is
uh,
like the rotation
yeah, yeah, yeah,
flap is
yeah,
like
yeah, exactly.
how they recover.
Yes.
But I don't know about like they can be used to be in nucleus and others.
Yeah, yeah.
Other.
In nucleus specifically, FRAP is one of the most used technique to actually assay this nuclear import. Okay, because this is a very cool technique to, you can photo bleach specifically the nucleus. Okay.
So sir, like if I can photo bleach the ER,
Huh?
I can then
Huh?
sense the modified proteins that are coming or that's outside the or like ER. They are
Modified?
rejecting the ER.
Modified?
Like we know that ER modifies and attaches some groups and other thing post transition modification.
Yeah, yeah.
So if we photo bleach the like except ER,
Huh?
I photo bleach the whole cell.
Okay.
Then what will be released from the ER? Will
Huh.
there be fluorescence?
Yeah, yeah. If you will photo bleach for whole cell, then of course, if your
Except the ER.
actually except the ear, you will photo bleach
Yes.
the whole cell except the ear. That is that I don't think that is possible, because
Okay,
uh,
so this contains small areas like very small portions
yeah, because
to be considered.
yeah, because excluding a small area and bleaching the rest of the area, that is very tough,
So
I
sir,
think.
like my question is now, how can you I mean tag the proteins that are modified in ER? Like we need to study about the proteins that are modified in ER. How can we recognize which one is the modified and which one
Okay,
is just
okay.
came
So you are
out from
asking
ER? And
that if you want to study that which proteins are modified by ER, right? Right.
yes, yes, yes.
Okay. So ER means modification means, as I can remember, that glycosylation, right? That is one of
Yes,
the
sir, glycosylation happens.
N-linked, O-linked that you were talking
Mm-hmm.
about, right? Okay. So you need to assay or you need to find that uh, that whether your target protein is being modified in the by the ER or
Modified
not.
or not?
Huh?
So, my target protein has been modified or it is not. It is in wild type. Like nothing happens to it.
Okay, so you want to see that whether your protein is modified or it is not modified, unmodified, right?
Hmm.
Right.
Yes, sir.
Yeah, yeah. So if you are asking about ER specific modification like glycosylation, so for that you can do a simple thing. Glycosylation, you yeah, you can do. See, you can just do a simple Western. Okay, and in Western you will do a lectin blot. Okay, because lectin is a lectin.
Attached to carbohydrates
Yes,
or
lectin has the
protein.
interaction affinity to the carbohydrate. So as your does the carbohydrate wala modification, glycosylation. So if your protein is unmodified, then your lectin blot you will not find anything. But if your protein is glycosylated, then you will find the the modification, or you will find the band by using the lectin blot. So that is one of the way which I know where glycosylation can be, you know.
And so, like we can use in lectin in chromatography and separate the proteins that
Ha
are
ha ha
glycosylated.
ha ha ha. Or
Okay, okay.
and the best best thing today what people do is just simple to the mass spec. If you will do the mass spec, everything you will find that
Mass
that
spectroscopy.
yeah, mass spec, you will find that which location you have the modification, that information you will also get. Okay,
Okay.
or if you don't have the modification, that information you will also have, because with this
Okay, sir. I
era.
need to study spectroscopy also. I didn't started yet.
Yeah, mass spec actually in research it is widely used. So mass spec
Yes, sir.
use
NMR and mass spec both needed. Like
mass spec mass spec generally needed for this kind of post-translational modification or sequencing, phosphorylation. So to find that specifically and the sequencing, so for that generally mass spec is being used. Yeah.
okay.
So for that you can do your mass spec. Or otherwise, the if you are talking about the biochemical technique, then you can do the lectin plot. That will
Okay.
also give you
And
a
about the any structure folding, we can use crystallography, right?
yeah, structure folding. You can do X-ray crystallography, NMR, cryo-EM, anything you can
And
do.
sir, cryo-EM, I just started studying in YouTube that we can sense the 3D structure of anything by cryo-EM.
Yeah, 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 yeah. So yeah, you can of course study about the cryo-EM, and yeah, now
Okay,
people are
sir.
being shifted to cryo-EM from X-ray and NMR. So the demand of cryo-EM is being increased now.
Will it helpful for us to study now?
Yeah, it will give you better resolution than, of course, than X-ray and NMR too. So that's why it is being widely used now. IEM. Yeah. Okay, so let's move on to the last slide, which you asked about mRNA export. So I will not just discuss about very brief. I will just have a brief discussion about the mRNA export. So here you can see the transcription is going on inside the nucleus. Okay, so now mRNA has to move out from the nucleus. So for that, as you know that this is the RNA polymerase, and this is the cap binding protein, and this is the 5 prime cap. As you know that in mRNA there is a incorporation of the 5 prime cap, right? So when it will be synthesized simultaneously, the cap binding protein will come here. Another protein HRNPC tetramer will come here, and this recruitment of this protein will actually lead to the processing of the mRNA. As we know the splicing, right? So it will help to the splicing, it will remove the introns. So after recruitment of this complex H and RNP, you can see that splicing has been occurred. So here splicing has been occurred. So now the mRNA has the 5' cap, the intron has been removed by the splicing, and now after that, after the splicing, the TREX complex and SR proteins will be recruited here, because that from this stage now the cell has been start to initiate the process of mRNA export. So one of the important protein is the TREX and SR protein. This is the red one is the TREX, this is the SR protein. So why their recruitment is required? Because upon their recruitment, the the mRNA export transporter will be recruited, which is the MEX667 and MTR2. These are this is a sorry, I think some problem. Yeah. So upon their binding, checks and SR protein, it will make a landing platform for this heterodimer protein MEX67 MTR2. Here you can see three molecules of this heterodimer has been bound here, and these are the and this is the mRNA export transporter. So after they bind here, and the polyethyl also has been added here. So now after that, this complex which has the MEX67 MTR2, this will bind to the NPC. This is the NPC, and they have the sequence which can interact with the NPC, and as you know that NPC has the FG repeat, phenylalanine and glycine. So these proteins MEX67 and MTR2, which are export transporter, they have sequence which can interact with the nuclear pore complex FG repeat, and which will make and which will mediate the export of the RNA through the nuclear pore complex. So that's why the release of the mRNA will be possible from the nucleus to the cytoplasm. So also for mRNA, you need to have some recruitment of the mRNA export receptor, which is MEX6 or mRNA export transporter, which is MEX67 MTR2. So here also you need to take help of someone, a transporter, to get out of the nucleus. Is it clear now?
Yes, sir.
Is it clear? Any doubt you have? Any confusion?
Yes, sir. I just confused because I thought that mRNA doesn't need anything, but it was written that mRNA doesn't need RAN GTP or.
Yes. That is true. mRNA does not need RAN GTP or the importin exportin, but mRNA needs this. By alone, it cannot be exported. It needs to take help from someone. Okay.
Yes.
Yeah. So this was the last slide from today. Okay. So my PPT is over. So so I'm asking you, if you have any question, tell me. If you have any doubt, otherwise we can, I think, close the session.
So sir, like we need to deactivate any protein in our cell that is formed in cytosol, and we don't need.
Sorry.
I mean, like there is some proteins in a cytosol synthesized by the ribosome, but we don't need them to be go inside the nucleus and express their and transcription factor, like activate anything. So we can design any type of proteins that can attach with the NLS and can mask them from attaching to the importers and import them inside the nucleus.
If you don't want to send your protein inside the nucleus, then you should not add any NLS there. If you don't want.
No, sir. Like some proteins are already been formed in our body. Like they are like already like gene expression.
Okay.
Some genes express and some misfolded proteins or anything happens like in a disease like. So we have to stop it to go to the nucleus. So we
Okay,
can
okay.
block the NLS by anything protein.
Yeah,
We
yes,
can
very
say.
good. That's a very good idea. You can you can design such kind of inhibitor which can bind to the NLS so that to disrupt the interaction between protein and the protein. So yeah, that's a very good
Sir,
idea.
we always need to be. It's also need to be protein to interact with the NLS, or we
No, no,
can
no, no.
design anything.
You can you can use drug also. Why only protein?
Drug
Your
also.
job is
So
to
we don't
just
know about drugs how interact with the proteins and
huh.
like DNAs or anything other things.
Yeah, sorry.
So how drugs are like interact with proteins and anything in body.
Same way, generally the drugs protein interaction is already it involves the non-covalent, mainly the non-covalent interaction, like two protein protein interaction happens. वही मतलब hydrogen bond, ionic interaction, salt bleach, hydrophobic
So like
interaction.
it blocks the space or anything that is required for its
There is
processing or.
a binding site for the drug to its target. So yeah, so by keeping on that mind, you need to design one drug so that the drug can
Sometimes it
fit.
inhibits and sometimes it can regulate it to express.
This generally it will inhibit. It will just sit on the binding site and it will know. It will not let your protein to do its work. Actually, it will just
Okay.
block the binding site. For that, you need to design a drug which has more interaction ability with your protein binding site. Then I mean, you can inhibit. Okay. Got it?
Yes, sir.
Yeah. Okay. Okay. So, any question? Any of you have any question or anything to be discussed?
Sir, why budding yeast is always used or yeast is used for eukaryotic, like for expression of genes and anything like process and everything.
Yeah, because budding is to grow. You can grow it very easily. It is follow all the eukaryote features, and you can grow it very
But it
easily.
don't have the all the genes and regulations we have.
Yeah, but for that cases, it has the bottleneck. Like if you want to do some intron study, you can do it. You can't do it budding yeast because in budding yeast there is very less amount of intron. So if you are studying splicing or so for that case you can't use budding yeast. So there are
So
some
like
bottlenecks.
limitations
Of
about
course, of course, limitation has,
study.
but the things which are similar to the other eukaryotes, for that study, budding is a very useful or very easy to handle organism and very fast. So that's why it is, if you have the option, you can use it. It is easy to handle and it is every 90 minutes it will double, right? So it will give you a more. I mean, it will become an experiment fast. Right.
And sir, one more thing.
Yeah.
During the centronemal complex formation, so the chromosomes align one above another, and then the strands are been like attached by SMC, or they stand side by side, and one of the sister chromatids are different sister chromatids, non-sister chromatids are attached.
They stand by. They actually align side by side, and then.
So like there will be attachment of one non-sister chromatids of each chromosome.
Huh, huh. One non-sister
So
chromatic
like
per each homologous pair.
uh, yes sir. So like during crossing over, one strand will be changed from the both non-sister chromatid.
Yeah.
Then after segregation, there will be one strand that is
Recombinant.
recombinant, and one strand like in homologous not sir, in one chromosome one sister chromatid is
Homologous,
recombinant, and one is not recombinant.
Yes, one recombinant, one non-recombinant, and for the second pair also one recombinant, one non-recombinant.
Yes sir. So So like if we see here that after the meiosis end, the one every cell has one strand only. Like
Your voice,
non-recombinant.
your actually your voice broke down in between, so I could not listen to
Yes,
you.
sir. Is it now
Yeah, yeah,
okay?
yeah. Yeah, is it visible?
So I just mean to that in the end step there are four cells. Two has recombinant and two has non-recombinant.
Yes, perfect. Yes.
But we told everyone that they are not identical because they are separate from parents. Like one stand from mother and one parent. So the non-recombinant also are genetically diverse because they come from different parents.
Huh. Huh.
Is that the reason?
Yes. 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 When the actually when the zygote will form, the zygote will have possibility that one yeah, one either one recombinant and one, there's possibility to have one recombinant and one non-recombinant, but these two non-recombinant and recombinant, whatever it will have, these two are from two different sources, right, in the zygote. So that will actually bring you the genetic diversity.
Yes, sir. Like they are from different parents and
Different pairs,
different
exactly.
organisms.
Their sequence is
Yes.
of course different, na? Their sequence is
Though
different.
they have not occurred through the recombination, but still their genetic diversity occurs
Yeah,
because
yeah,
they have
yeah, yeah.
separate parents.
Yeah,
Okay, okay.
separate parents. Yes, yes. Okay, crossing over will just increase the level of genetic diversity, and that it will do. Okay. Okay. So anyone else have any doubt, or anyone of you? Is it clear to everyone?
Yeah, sir.
Okay. Okay. So if there is no doubts, then I think I should close the session. Okay. Then see you on the next week.
Yeah, sir.
Okay.
Thank you, sir.
Bye. Yeah. Most welcome.
Bye, sir.
Bye.